So this video will provide some additional information about the Unknown Project. So just to give you an idea of how the Unknown Project would work is that you would be given a broth, and in the broth that would contain two types of bacteria. You would have 1 gram positive and 1 gram negative. And so both of those bacteria would be mixed together. And you might recall back when we talked about how we isolate bacteria from a large mixed culture, you might recall that we did our streak plate, right? A streak plate is used to isolate individual colonies of bacteria from a large mixed population. And so when you are given the broth, you would perform 2 streak plates. One would be on a nutrient agar plate, and on your nutrient agar plate, both bacteria should grow because that media is very general. It supports the growth of many types of bacteria. So on your nutrient agar plate, when you do your streak plate, you should be able to see 2 colony types. There should be 2 different colony types on your plate because 1 colony type is your gram positive and 1 colony type is your gram negative. The second streak plate that you would do would be for what's called a McConkey agar. Now, you need to read in your book about the McConkey agar. This is in activity 4-5. And so you're going to need to read that section to understand the purpose of using the McConkey agar. Now, on the McConkey agar plate, only 1 type of bacteria should grow. It's not going to grow both your gram positive and your gram negative. Only one of those will grow on the McConkey agar. And so when you do your reading about it, you'll be able to figure out which bacterial type grew on that agar. So when you look at your McConkey agar plate, there should only be 1 colony type. There's only going to be 1 bacteria that grows on your McConkey agar. And so let's talk about what we would write up for our readout. So let's say that this is your nutrient agar plate. So I've already given you instructions for how to go about finding your nutrient agar plate, but let's say this is your nutrient agar plate. Remember that this plate is going to have 2 colony types because you have your gram positive and you have your gram negative. So what you want to do is you want to look at your picture and you want to try and find the 2 colony types. So I have colony type A, so I'm going to call that one colony type A, and this is going to be colony type B. So these are my 2 types. You can actually see it pretty well when you see the colonies side by side. And so this colony type is different than this colony type. So these are my 2 colony types. They're not necessarily different colors. They don't have to be red and white like we saw in our streak plate because it's all dependent on which bacteria are in those media. So in this case, these are my 2 colony types. So when I write up my ID sheet for this nutrient agar plate, I want to describe my 2 colony types using figure 2.3. That's in activity 2-2 in your lab book. So in the environmental culture, you're going to find some terms you can use to describe your colonies. So this is an example of those pictures. And so you're going to do your best. Now, obviously, you're not in person. It makes it a lot more difficult to say the elevation, let's say, because you're not looking at your plate from the side. But you could describe it in terms of the margins. Are the margins smooth and entire? Is the colony round? Is it, see how this one, you can see kind of this raised portion? That would be this Umbani type colony, etc. So you want to try and describe your 2 colony types to the best of your ability. You could describe the color, whether it's more opaque or transparent or solid white or cream colored. You want to use a variety of different terms to describe your 2 colony types so that you can distinguish between your 2 types, right? So I have 2 types of colonies and I need to describe both. So I will have an identification sheet for my nutrient agar plate. And so on my nutrient agar plate, I'm going to put that picture in the test results. Now, I also would recommend, just like I did in the slide before, Label your 2 colony types. So you could see in the picture, I labeled the 2 types. So label your 2 types. So this is one colony type. This would be another. And so I would actually label this and then put that like colony type A and colony type B. So that's for that. Now, name of the test, streak plate on nutrient agar, purpose of the test, right? So what is the purpose of doing a streak plate test? And specifically, you want to add, why are we doing a nutrient agar plate? What does that allow us to separate? And then in terms of our procedure, I'm going to come back to this in just a minute. That is going to be your steps that you did that basically was how you did your experiment. And so I'll come back to that in a minute. You're going to include your picture. So this is my picture from my streak plate. And then for your interpretation, the interpretation for your streak plate is going to look a little bit different than what's listed here. This is more for your biochemical tests. For your interpretation section, this is where you're going to describe your 2 colony types. So you would indicate colony type A has colonies that are umbani, smooth entire edges, they're larger, they're more transparent, they're creamy white. Whatever terms you're going to use, that's what you're going to put in the interpretations. So colony type A looks like, and describe that colony. Colony type B looks like, and describe that colony. That's what you want to put in your interpretation section for the nutrient agar plate. You basically just want to describe, what do your colonies look like? Now, in terms of procedure, that's going to be the next slide. So you might recall back when we did our streak plate, you have an image that looks like this. And this basically tells you the steps in your streak plate. So when you're describing your streak plate, right, you're going in your steps, vortex the tube to mix the bacteria in the broth, right? So vortex your bacteria. Remember that you don't use first person or third person. You don't say I did. It's just the steps. So vortex tube to mix well. Take your loop using aseptic technique, dip the loop into the broth, and then draw a line down and zigzag inoculate into the first quadrant. So that would be a step. The next step, flame your loop, let it cool, make 6 to 8 directional streaks. Flame your loop, let it cool, make 6 to 8 directional streaks. And then the next step, make zigzag streaks coming from the third quadrant out and zigzag into the open space. Now, don't forget, it's really important that the last step of this procedure is to tell somebody how you would incubate this plate. So incubate the plate at 37 degrees Celsius, agar side up for 48 hours. Most of our tests, the time is going to be 48 hours. So incubate your plate, agar side up at 37 degrees Celsius for 48 hours. That would be your steps for the streak plate. Now, you don't have to go into all of the detail in terms of aseptic technique. So again, I mentioned that I do aseptic technique, but then I don't state all of those nuances. Like I don't have to say, flame your loop, let it cool. Take your tube and put the cap between your pinky and ring finger. Open or remove the cap from the tube. Flame the opening of the tube. Stick the loop into the broth. Flame it again. You don't have to go into all that. Assume the person knows aseptic technique. You do need to say the part, though, that matters, which is the technique in terms of how to do your streak plate. And then you also need to include the step for the incubation. Now, for the McConkey agar, again, you need to read activity 4-5 because one of the things that you're going to see, and again, this is going to go with our endo-agar and our membrane filter technique, is that we're starting to look at media now that is both selective and differential. And so I would recommend going and watching those videos first. So watch the video on the membrane filter endoauger video. That's one video. And in that video, I talk about selective ingredients and differential ingredients so that you start to see what does that mean. Basically, what you're going to see, though, is when we talk about selective media, it has ingredients that selects for only one type of bacteria to grow, meaning it has ingredients in it that inhibit other types of bacteria from growing. And so you need to understand, what are the selective ingredients in the MacConkey agar? What are the differential ingredients in the MacConkey agar? And so that's going to influence the way that they look on the plate. So notice I have E. coli here. I have Enterobacter aerogenes. Notice when they're growing on MacConkey agar, the plate has this pinkish red color. That tells us something. Proteus vulgaris, Salmonella typhi, they grow, but notice they're not red. They're not that pink red color. And if I look at Staph aureus, for example, that bacteria does not grow on MacConkey. And so there's a reason that this bacteria does not grow. So you need to do some research about the MacConkey agar so that you understand what the results mean when you look at your MacConkey agar plate. And so let me give you an example. So let's say that this is my picture for my MacConkey agar plate. So again, when I make my ID sheet, I'm going to have my title. So streak plate on MacConkey agar. Then I'm going to have the purpose. So read through the instructions for the MacConkey agar. Why do we use MacConkey agar? So the purpose, the steps are going to be very much the same as they would be for the streak plate for the nutrient agar. So the procedure should be very easy. In terms of the results, you're just going to include your picture in that result section. Your interpretation is going to look a little bit different though. So in the nutrient agar interpretation, you're describing your two types of colonies. However, in the MacConkey agar, only one type of bacteria is going to grow. Again, I'm not going to tell you which one. You have to read about that type of agar, but only one of your two organisms will grow on the MacConkey agar plate. So when you are talking about the interpretation, so when you're interpreting your plate, in order to get full credit, you need to discuss the selective ingredients. So what are the selective ingredients in the media and what that tells you about what type of bacteria grew on the plate? So what are the selective ingredients and what does that select for? What type of bacteria does that inhibit? What type of bacteria is allowed to grow on this plate? So discuss your selective ingredients and what that tells you about bacteria that are growing on this plate. The next part, discuss the differential ingredients and what the color of the agar tells you. So the fact that this one is pink tells me something about this bacteria. And so in my interpretation, I need to describe what that color tells us. Now, if you look at the drive that has the MacConkey agar plates, you'll be able to see different images that are in there. And so if you're struggling to tell if those colonies look pink, compare your plate with other plates. That sometimes will help you distinguish, like this one is very clearly pink. That tells us something about this bacteria. It tells us about a certain food source that they use. And so in your interpretation, you would need to talk about that. What are the differential ingredients and what color does that make the agar and why? So for your MacConkey agar, this is the information that you need to put in your interpretation. Now you need to talk about selective ingredients and differential ingredients and what does that tell you about the bacteria that's on that plate. So that's for your MacConkey agar. Now, when you make your ID sheets for your nutrient agar plate and for your MacConkey agar plate, remember that you're going to upload those two ID sheets into Canvas. Then you're going to submit them. That's going to be graded. After those are graded, then I'm going to give you the next part of your experiment. So if we were doing this in class, you would actually do your streak plates, and then 48 hours, you would get your plates out, and you would use those plates to do the next part. And so the next thing that you would do would be to do a gram stain. And you have to gram stain from the nutrient auger plate. There's 2 reasons it has to be from your nutrient auger plate. One is you want to gram stain both colony types. The McConkey auger doesn't allow both colony types to grow. There's another reason why you can't gram stain from McConkey, and when you read about McConkey, you'll understand why you can't gram stain from that plate. So your gram stain would have to be from your nutrient auger plate because it has your 2 colony types. And so when you do this, what we would have you do is you would actually start with 2 water puddles. So this is different than when we did the demonstration for the gram stain, where I had 1 puddle of water and I basically mixed the 2 bacteria into the 1 puddle. This is different. I don't want to mix them together because I want to know for this colony type, is that one a gram positive or a gram negative? And then for this colony type, is it a gram positive or negative? So to do that, I'm actually going to have 2 circles of water. I'm going to have 2 circles of water that I add using a loop, and I make those circles of water again about the size of a nickel. I use my needle and I pick up my bacteria. So I would pick up colony type A, and using my needle, I would swirl it into that water puddle. Then for colony type B, I would use my needle, swirl that into this water. I would air dry my slide, and then I would do my heat fix step. And then I would do my gram stain. So make sure that when you're talking about the procedure, that you're including all of that. How do you make your slides? So describe how to make a heat fix smear. Also describe how to do your gram stain, the steps that would be used in the gram stain. And so in your report, when you're writing up your actual report, there is a section that says, why do we gram stain on the same slide, but side by side? So I want you to think about, why would I not want to have one on one slide and one on the other? Think about the most important step during the gram stain procedure, the step that's going to make that procedure vary. And could doing them on 2 different slides cause a problem? So there's a reason that we would do them on the same slide side by side. And so you need to think about that. And that's going to be written up in your report as to why do we do the gram stain side by side. So again, not mixing them together because then we don't know which is which. Side by side, but on the same slide. So when you submit your plates, your ID sheets for your nutrient auger plate and your McConkey auger plate, then I'm going to give you back in the comment section pictures for your gram stain. You're going to have 1 gram positive and 1 gram negative. And so when you're looking at these, I'm going to give you these pictures. I'm not going to tell you which one is which because I want you guys to recall which color is which. But you are going to use those pictures and then describe the type of bacteria that you have. So for your gram negative, I'll give you a hint. For your gram negative, you don't have to worry about arrangement. But for the gram positive, if there's an arrangement, you need to indicate that. And what do I mean by arrangement? Well, again, remember we talked about staphylo or strepto, right? So strepto would be in chains. So notice like this, these are in chains. So strepto would be an example. Staphylo would be an example if they were grape-like clusters, so on and so forth. So when you're describing these, you're going to describe morphology, shape. So are they rods? Are they coccus-shaped? Are they spirals? What is their morphology? And what is their arrangement? And so let's talk about what would go on your ID sheet for your gram stain. So this would be like what you would have for your ID sheet for the gram stain. So your name of the test, gram stain, you're going to write the purpose of the test. You're going to write the procedure that is followed, right? So again, talk about how you make your heat fix smear, from which plate are you using, and then also describe the procedure for the gram stain. Test results, you're going to indicate or you're going to put in the pictures that I give you. Those are the pictures that need to be included in the test results, right? So I need to include both of my pictures here. And then in terms of interpretation, the things that you want to include in your interpretation for full credit, describe your 2 different bacteria in the gram stain. So both morphology and arrangement. So where you can say that. So again, arrangement is more for the gram positive, not as much important for the gram negative. So you're going to describe morphology and arrangement. You are going to indicate if endospores are present. So think about if we were to do a gram stain, first of all, you need to think about, would that be in your gram positive or gram negative? So you might have to go back and review for the gram stain, or I'm sorry, for the endospore stain, if endospore producing bacteria are gram positive or negative. So it's going to be typically one of your 2 types if you have endospore producing bacteria. Not everybody does. But if there are endospores present, right? Notice we're doing a gram stain. There's no heat in the staining procedure part. There's no heat when we add our stains. So you should think about, what would that look like if I were to gram stain bacteria that has an endospore? And so you want to think about what that looks like. And so that will help you figure out, do you have a bacteria that is an endospore producer? You want to include in your interpretation which color is gram positive and which color is gram negative. So the gram positive bacteria appears blank, while the gram negative bacteria appears blank. And in there, you want to describe, why do gram positive and gram negative stain differently? Be specific. So why is one purple and why is the other one pink? So describe why they stain differently. And then the last thing you're going to do is you're going to propose a genus for your gram positive bacteria. So based on the morphology and the shape and the arrangement, you would then propose a possible genus. So let's say for my gram positive, I have caucus shape, so spheres, and they're in chains. Well, I could propose that my genus would be Streptococcus, right? Streptococcus is a gram positive, and it's spheres that are in chains. So you want to think about, what possible gram positive genus could you have? If they're endospore producing bacteria, you need to think about what genus that could be, right? We talked about that there are 2 main types of endospore producing bacteria. There are 2 main genus. Now, the way that we grow these plates is going to be in the incubator, right? It's in our incubator. So you want to think about, because that's in the incubator where oxygen is present, that is going to rule out one of our 2 genus of endospore producing bacteria. So make sure you keep that in mind when you propose the genus for an endospore producing bacteria. The way we grow those plates is going to basically rule out one of those genus. So you don't want to say that one, because it's not possible if we grow it in the incubator. So propose a genus for your gram positive. Now, for the gram positive, this is as far as the identification goes. You are not going to do any further tests for the gram positive. The identification stops at your gram stain. You're just going to propose a genus, and that's it. Your gram negative, though, you are going to identify all the way down to the species of bacteria. And so in addition to the gram stain, you would also set up an Invic. And so you would take your McConkey agar plate and you would use that plate to inoculate an Invic. Now remember, for an Invic, we should have 3 tubes. We have our tryptone broth, we have our MRVP broth, and we have our citrate slant. Notice you do not need 2 tubes like we did in our Invic. In our Invic, in the video that I described how to do an Invic, there were 2 bacteria that we tested. We tested E. coli and we tested Enterobacter aerogenes. That's why we had 2 of each tube. In this case, we're only testing 1 bacteria. We're testing the bacteria that's on our McConkey agar plate. So you only need 1 of each tube. For this section, this will have 4 ID sheets. You're going to have 1 identification sheet for indole, 1 for methyl red, 1 for Voges Proskauer, and 1 for citrate. So this has 4 different identification sheets. Put them on separate sheets. Make sure to include the steps for the inoculation. So how do you inoculate these broths? You also want to include your incubation steps. So incubate in a 37-degree incubator for 48 hours. And then don't forget the steps after the incubation if the reagent is used. So like for the tryptone broth, for example, when you take the tube out of the incubator, what needs to be added to that tube in order to have your readout work? And if you're talking about the MRVP broth, remember that this is a 1 broth and it's used for 2 tests. It's used for methyl red and it's used for Voges Proskauer. So don't forget that when you take this tube out, what do you need to do to test it for methyl red and to test it for Voges Proskauer? So make sure that you include those steps as well. After you get your results for your Invic, you will then use that information to find your flowchart and then request the next test. So again, when you submit your nutrient agar and your McConkey agar plates, I am going to give you the results for your Invic. So I'll give you the pictures for your 4 tests. I will also give you your picture for your gram stain. So you're going to write your ID sheet for the gram stain. You're going to write your ID sheet for the 4 tests for the Invic. You're going to submit all 5 of those together. And when you submit it, you're going to tell me which test you need next. So let me show you what I mean by that. So on Canvas, there are what are labeled flowcharts. And so you'll open up a document and it looks like this. And so you're going to find your Invic results. So based on the pictures you get, you're going to find which one of these figures goes with your result. So let's say I look at my Invic results and I got indole negative, methyl red negative, VP positive, citrate positive. So negative, negative, positive, positive. That means that I need to be on figure 1. So I would scroll down to figure 1 and it looks like this. So again, negative, negative, positive, positive. So when I submit my ID sheets for my Invic and my gram stain, in the comments section, I would need to say, Dr. Wells, I need the result for lysine decarboxylase. That's the next test. So Dr. Wells, I need the result for lysine decarboxylase. And in the comment section, I'm going to reply and I'm going to give you the picture for the next test. So I will give you your result for your lysine decarboxylase. So let's say the picture I gave you, the tube is purple. Well, if your tube is purple, you're going to write up an identification sheet for lysine decarboxylase. That's the next one you're going to write up. You're going to include the picture that I gave you that shows that it's purple. You're going to fill out the ID sheet in terms of what does that interpretation tell you. And then when you submit this ID sheet, Then you would ask me, Dr. Wells, can you give me the picture for motility? And then in the comment section, I would give you the motility picture, and the process would continue until you get down to the species. So notice that these series of tests are going to allow me to identify which species of gram-negative that I have. All of the bacteria that are on these flowcharts are gram-negative. So I will be able to figure out which of these bacteria is my gram-negative. I would be able to identify all the way down to the species. And so this is how I would do my unknown project. So I need to start with my nutrient agar plate first and my McConkey agar plate first. I need to streak for isolation. After isolation, then you can take your plates and you can set up an Invic and a gram stain. Then you would get your results for your gram stain. You would get your results for your Invic. After you get your results for your Invic, then you would set up the next test. Then you would get the result from that test, and then you would set up the next test. And you would keep doing this until you identify what bacteria you have. So all of those biochemical tests that you've learned throughout the semester, the purpose of doing those tests is to help you to identify what bacteria you have, because we know certain bacteria have different reactions, like that Klebsiella pneumoniae is going to be negative for motility, and that Serratia marcescens is going to be positive for motility, and so on and so forth. So all of those biochemical tests that we've done are basically used to help us to identify unknown bacteria, because we can do these series of tests to figure out what bacteria we have in our original broth. And so those are things to keep in mind when you're doing your unknown project. And so hopefully this clears up some confusion about what needs to be addressed in your unknown project at the beginning.